right, well, today's lecture on the eye. Let's get started. Let's see here. A couple pictures of the eye here. The picture on the right we kind of looked at for the extraocular muscles. The picture on the left is what I want to teach now. It's called the Lacrimal Apparatus. It's responsible for wetting the eye with your tears and draining them. So, okay, let's kind of draw what we see here. We see the eyelids enclosing the eye, part of a bigger eyeball. The lacrimal apparatus includes a gland called, this gland kind of up here, laterally and superiorly. That's the lacrimal gland that produces the tears. Kind of in this area here, glandular tissue. And when it's stimulated, it'll produce tears, teardrops. Now, when you blink, it sweeps the tears that are produced across the eye from lateral to medial to this location here, kind of towards the medial eye. This location where the tears are swept is called the lacrimal lake. Actually, let me write the term over here. Draw some more stuff on this side. But that being said, you need a drainage system when the tears are swept from lateral to medial. And so you have two little orifices on the picture there. They're very, well, you can see them, the pointing right here and here. They're the superior and inferior puncta, little orifices to drain tears. Superior, inferior, puncta. A little orifice there and there, just put little dots to signify where they might be. And then you need a drainage system to drain those tears. So you have these little canaliculi, superior, inferior, lacrimal canaliculi, here and here, that drain the puncta. So they're going to drain the tears into this dilated sac called the lacrimal sac. Think of the lacrimal sac as kind of the widened, dilated portion of what becomes the nasolacrimal duct. Normally tears are drained down here, as you can see from the picture, in the nasal cavity, right by the inferior nasal concha. That's basically it. These structures I've listed here are the tear producers and drainers called the lacrimal apparatus. So that's why when you have an excessive production of tears when you're emotional, you get the sniffles, because it does drain in the nasal cavities. All right, so I want to move away from that and talk about the eyeball itself. Today we'll have the eyeball dissection lab, and you'll remove the eyeball, and I just want to show you the basic parts. You'll have to do some handiwork to remove all the fat and extraocular muscles to get it down to the white-looking eyeball. And actually, there are many layers, 3 layers of the eyeball that we define. They're called tunics, layers. The fibrous tunic, see if I can draw this. Okay, well anyways, I just drew the outermost layer. I use black. It's colored white. It's the whites of your eyes. The fibrous tunic consists of, well, pretty much any part that's known as the whites of your eyes is considered the sclera of the fibrous tunic. Whites of your eyes. Basically, this provides an attachment for extraocular muscles. It's the tough part. Now, also part of the fibrous tunic, more anteriorly, I draw it right there, that's the lens. Or not the lens, the cornea. Well, the cornea works with the lens to bend rays of light so you can see objects clearly and in focus. It's mostly the lens that helps bend the light rays, but the cornea, you can bend light rays too. Think of the cornea as the window of the eyeball. Light first must pass through there, the window of the eye. Lets light rays through. If you ever had LASIK surgery, that's kind of what they manipulate. They kind of shave off layers to help increase depending on the situation. Bend the light rays a little more. Maybe you have a super strong prescription. Maybe you're a good candidate for LASIK if the cornea is more flat. It gives them more leeway. They don't have to shave off as much to bend the light rays more. If your cornea is very round, my understanding is you're not as good a candidate for LASIK. They would have to shave off too much cornea to bend light rays a little. It's advantageous to get LASIK if you have super strong prescription, you need contacts and glasses, and it's very, corrective lenses are difficult. But if LASIK can really weaken your prescription, make it so you need less help, make your eyes stronger, it might be advantageous. Or maybe you don't need corrective lenses at all. Okay. If you don't mind corrective lenses, I would just wear corrective lenses. Don't let them mess with your cornea. What if it doesn't work out? Can't get that stuff they shaved off back. Anyways, cornea. Okay, any questions on fibrous tunic? It's pretty basic. All right, well, internal to that is a dark layer called the choroid. Well, it's the vascular tunic. The choroid is a part of it. It's a dark layer there. It includes the choroid. Choroid is complex network. In this case, it's described as a vascular bed. You know, blood vessels. So I'm going to use red to symbolize that, this layer, the vascular tunic. Okay, that starts here. Inner layer. Okay, so I drew a bunch of stuff there, and all the structures are listed there. The layer itself, right there, that is the choroid, okay? The vascular bed. However, if you go anteriorly, there's some other structures, like the ciliary body and the iris. Right there, that's the ciliary body. The ciliary body contains muscle, smooth muscle, that we talked about, that's innervated by cranial nerve 3. That helps focus the lens. That's what we talked about. Okay. When the muscle contracts, it basically allows the lens to bulge, as I was saying earlier. So there is a lens behind, well, first let me finish describing the parts. So we got ciliary body. This part here is the iris. Just that right there. Colorful iris. Okay. We said that the iris can constrict or dilate. And I taught you the sympathetic, parasympathetic function. Cranial nerve 3 is the parasympathetic function, and that was for, do you remember? Constrict. That's the parasympathetic response. Sympathetic, dilate your pupil. So the iris has a little space right there. The space that actually light has to go through is the pupil. However, you also see this pretty arrangement. 360. You'll see it on your cow eyeball much better. It's called the oral serrata. Ora serrata. I didn't draw pretty well. But anyways, I wanted to leave some space to draw the lens, which is right behind the pupil. Okay, that's the lens. The lens is held in place by some suspensory ligaments. Okay, so those lines holding the lens, suspensory ligaments. So there are some important structures in this tunic. You got the blood supply, the vascular tunic. You got the choroid, ciliary body that helps you focus the lens. You can constrict or dilate your pupil. Okay. The lens will help you see far or near, okay, depending. So when you let light in, you have to have cells that can actually absorb the photons of light so you could see. And those cells are called photoreceptors, and they're in the neural tunic, the innermost layer. It's colored yellow on the figure. I don't have yellow, I'll use orange. Hopefully it's not hard to distinguish the orange from the red there. Anyways, if you look here, there's a space in the middle for the blood vessels to emerge inside. I'll just draw some arteries and veins coming in there for the blood supply. But pretty much, that's cranial nerve 2 coming out the back of the eyeball. You should be able to see that on your cow eye. Cranial nerve 2, optic nerve. Pretty much this orange part. Cranial nerve 2, the optic nerve. You know, as the nerves go inside, they form the neural tunic. There's a couple of points here. One point where the blood vessels come in, where the optic nerve kind of comes in and spreads around, there's no photoreceptors there. So they call that the blind spot, or the optic disc. This term here, optic disc, known as the blind spot, because there are no photoreceptors in it. You have no photoreceptors, there's no signal to be sent. Those are the cells that can bind the photons in light, but they're not there. Your highest concentration of cones, actually, there's a little point right off to the side there. I'll just draw it as a little black dot. It's very difficult to see. Many of you may not find it. It's called Macula Lutea. And right at the center of that dot is the fovea centralis. Okay. So I'll put a little circle around the dot. The circle, which is supposed to be yellowish, macula lutea, but the dot in the middle, fovea centralis. Sometimes it's just called the fovea. The full name is fovea centralis. You have your highest concentration of cones in the fovea. Cones, I'll teach you later, they're for color vision and acuity. You can see sharp things. That's actually where you want to focus. When you look at something, you want to focus the light rays right perfectly on the fovea. That's where you have your highest concentrations of cones for color vision, acuity. Okay. So when you look at anything, look at someone, look at any object, when you see it clearly, it's focused on your fovea. Anything off your gaze is peripheral vision. Okay. All right. The rest of the orange layer outside of the fovea is just retina, where you have photoreceptors throughout, just not the blind spot. So call any, I don't know, point to some random place. Retina. It's the retina that has the photoreceptors. Okay. So when you get an eye exam, this is what the optometrist wants to see: a normal looking retina. A couple ways they can do it. The way I like to do it when I go to the eye doctor is I have to pay for it. It's like $40, $50. They do a computer scan. And they get that view. They're trying to look at your healthy retina, see if there's any problems developing. The other way, the old school way they used to do is they used to give you those eye drops to dilate. You have to walk around with those funny sunglasses all day. I hate that. I don't do that anymore. But what they're doing is, by opening up the iris, you get a better view of the retina. Okay. So what you see here, it's a little brighter at the optic disc. It's brighter where there's no blood vessels. It's a little darker, so it doesn't project well. The little dark spot is supposed to be the macula. And they also look at the arterial blood supply there. This is a normal looking one. Your eye doctor can tell if there's problems. For example, one problem that could occur, a common problem is glaucoma. There's fluid that circulates here in the anterior chamber. If there's kind of a blockage, the iris will kind of bend in a way that doesn't allow the aqueous humor to flow freely, so pressure builds. When pressure builds, it kind of exerts a pressure posteriorly. It can affect your vision because you're compressing the retina. So there are different things that you can look for in a retinal exam. I don't want to get into it. But one thing I do want you to know, since I said there's a blind spot, is if you were to print this out on a piece of paper, the plus and the dot, if you cover one eye and then kind of focus on the plus, at some point in moving the paper back and forth, the black dot disappears. Because at that point, the light from the black dot is on the blind spot, so you can't see it. Normally your blind spot is covered by the other eyeball, but if you only got one eye to look with, then the blind spot is in play. Okay. I mentioned the aqueous humor, so let me teach you the eyeball chambers and cavities and conjunctiva. So things to add to our picture include the anterior chamber, on the figure, I draw a little line between lens and iris from here to here. It's called the anterior chamber, the space basically between cornea, iris. Kind of hard to see. I tried to indicate the posterior chamber as being the space between lens, iris. Here to here. Posterior chamber. Well, anyways, those two chambers are what are known as the anterior cavity, where aqueous humor is continually replenished. So these two chambers, you can title it anterior cavity. Those two chambers are part of the anterior cavity. This is where aqueous humor circulates. And is continuously replenished. We don't have to know how it circulates, but like I said, if the circulation is impeded, pressure can build, causing glaucoma. Well, anyways, posterior to the lens, we call that posterior cavity. Okay. Pretty much, it's a big space. And it's occupied by the vitreous body or vitreous humor. I'll just draw a structure. That fills the rest of the eyeball, posterior to lens. Interestingly enough, this big gelatinous mass, called the vitreous humor or body, it's not replenished, it's relatively stagnant. And when you cut your eyeball in half, you should see a plop of jelly in there, or it usually disintegrates into liquid. As you age, it kind of liquefies a little bit. So post-mortem in a cow eyeball, I don't know what you see. You have to cut your eyeball open and see what's there. Expect fluid to come out. Oh, that being said, it's fluid-filled or jelly-filled. I've seen students puncture the eyeball in a way where you apply too much pressure. Maybe your instrument is too dull, but when you actually puncture it, stuff squirts. So eye protection, okay? Don't make it squirt. Cut it in a way where it doesn't squirt. It's not good. So this green stuff. Vitreous body, or just call it humor. Not the same as the aqueous humor where it circulates, it just kind of stays there. It provides intraocular pressure, which needs to be normal for normal eyeball function. So when the muscles pull in the eyeball with the appropriate pressure, it doesn't like deform the eyeball. You have an appropriate amount of pressure in there. Usually they test for that in the eye doctor. They put a little puff of air. It's kind of uncomfortable, but that allows them to measure the pressure inside your eyeball. Okay. Over time, if you get inflammation anywhere inside here, and byproducts of inflammation can cause what are called floaters, kind of like seed spots or stuff in there. Anyways, those can be removed. I'm going to move all. Let's start to look at what's in front of the cornea there. The conjunctiva is a membrane that creates a little pocket for your contact lens. All right, there, a pocket. So let me kind of illustrate it there. Conjunctiva. Draw a little pocket here too. It's basically lining inside of your eyelids and part of your eyeball to create that little pocket or recess right there. But anyways, what I drew in the pink line, that's conjunctiva. It's a clear membrane. Except when you get your bloodshot eyes, it's the, I guess the blood vessels get too dilated. Dilated blood vessels. For bloodshot eyes. Well, what can do that? You know, what happens when you get bloodshot eyes? You're allergic to something. Dust, smoke, maybe too much alcohol, right? So you just take some Visine. Visine is basically a vasoconstrictor eye drops that can kind of get rid of your bloodshot eyes. It can get infected, conjunctivitis, commonly known as pink eye, very contagious. But anyways, conjunctiva, part of the eyelid. I'll just draw some eyelid around here, covering the eyeball. Some eyelashes. In your cow eyeball, you'll see the eyelashes, and you can remove the eyelid and get rid of it. Moving on. So two pictures side by side. One is the eyeball by itself, and then with the eyelids on it. And I did teach a muscle inside here. Do you recall? Cranial nerve 3 innervates it, lifts your eyelid, levator palpebrae superioris. Easy to remember. Levator means open or close your eyelids. Open with a pupil eyelid. Palpebral refers to eyelid. Superioris. Top eyelid or bottom eyelid? Top eyelid. Okay. Well, anyways, they show a chunk of muscle in there. Pretty nasty, right there. Okay, anyways, moving on. All right, so now we kind of have a basic understanding of the anatomy of the eye. Let's talk about the cell physiology of the photoreceptors. Called that because they absorb photons of light. You take physics, you learn all about light. So I think I can erase this now. I like this picture that's showing to you because it shows the photoreceptors as one layer of cells. There's 3 layers in this neural tunic here, but it's kind of up there. Squashed right there. They can see those 3 layers when you visit the eye doctor. Anyone have their sights set on optometry school? Maybe? No? Well, here's a close-up picture of those 3 rows of cells. You have this pigmented layer. Okay. But parts of these cells called photoreceptors are stuck in it. I won't try to draw the pigmented layer, but let me draw the photoreceptors. There's 2 main kinds. There's rods and cones. Rods have this funny external segment, what are called rods. It's a body. Little discs inside that have the visual pigment. Well, anyways, rods. I'll draw a few of them. Rods do outnumber the cones, so I'll draw 1 cone. The external segment is more cone shaped. Rods and cones are your photoreceptors, rods and cones, named for the shape and appearance of this external segment. Photoreceptors have the visual pigment that can absorb photons of light. I'm writing here, have the visual pigment that can absorb photons of light. Okay, well anyways, they communicate with this middle row of cells called the bipolar cells. Right here. considered like neurons. They're going to eventually communicate with cranial nerve 2. Well, anyways, it's that innermost row, the ganglion cells, that actually have fibers that form cranial nerve 2. Okay? These other cells, don't worry about them. The aberrant cells, don't worry about those. Do study photoreceptors, bipolar cells, ganglion cells. So there's a fiber from the ganglion cells. They draw that, we'll draw it that way too. These are the ganglion cells in black. Well, anyways, I just drew two. Basically, those fibers form cranial nerve 2, sensory for vision. Okay, so those are the cells that we're going to study, in particular, the photoreceptors. So that's the arrangement. In the neural layer, retina. You ever get red eye in photographs? I think that's the pigmented layer, I believe. Well, anyways, the photoreceptors, one thing I should have pointed out before I advance the slide is the pathway of light, the pathway of the nervous signal of vision. Light has to pass through everything to get to the photoreceptors to trigger a cell physiological response. Then, if you do, the pathway is sent the opposite direction to cranial nerve 2. Okay, so light goes one way, the signal goes the other way on this figure. Okay, moving on. This just shows photoreceptors, 2 rods and a cone, basically. And here's a real picture. I like this picture on the left because it shows, well, you tell me, are there more rods or cones? Rods. Okay. What you see are the external segments. Cone, bunch of rods, but where do you have the highest concentration of cones? I already mentioned it. Fovea. Okay. Well, I like this picture here because it shows the regular cell arrangement of the photoreceptors. Outer segment, inner segment, synaptic terminal. I'm going to erase this picture. Outer segment of a cone. Outer segment. That's where the visual pigment is. Okay, that's important. Got these discs inside. Then there's just a regular cell body. They call it the inner segment. That's just like, you got your regular cell organelles in there. Nucleus, mitochondria, some other bodies in there. Endoplasmic reticulum, all that stuff. Anyways, signal transmission here, synaptic terminal. You have to communicate with the next cell, which is bipolars. Okay, I won't draw the cone, but you get the idea. There is a difference between rods and cones there. Notice how A rod is a true rod with discs inside. However, not so for the cones. It's just a folding of plasma membrane. It doesn't have discs inside. Plasma membrane is just highly folded. Visual pigment is there. Okay. Phototransduction is the study of, well, you got to study the visual pigment, which is, let me read from my slide, detection of light by the eye is possible because of a photopigment called rhodopsin, which is opsin and retinal, vitamin A. So that big kind of dumbbell-shaped thing, that's the protein part, and that's opsin. That skeleton key-looking thing on the inside, that is called retinal. That's rhodopsin. Okay? So it turns out there's different kinds of pigments. Each type has a different form of opsin. They have a sensitivity of different wavelengths. The visual spectrum, well, the entire electromagnetic spectrum, I do believe they teach that in chemistry, right? Gamma rays, X-rays, the frequency, they're very powerful, strong rays that can damage biological tissues. That's why you wear a lead chest protector when you go and get X-rays of your teeth. Well, anyways, when you get to the visual light spectrum, we can see that at larger wavelengths, radio waves, microwaves, infrared, can't see them. You can only see this small window of the visible light spectrum, which is a wavelength of around, 400 to 700 ballpark. The cones are named for their peak activity based on wavelength of light. Light is described by physicists as it's like particles that act like a wave. It's kind of, I don't know if they've really decided, is it a particle or is it a wave? But they definitely have both properties. So blue cones work at a peak of around 420. Rods are around 500. Green cones, 530. When you're in the red, it's about 560. So it's a very, very narrow range. So I'm just running into the visible light spectrum is approximately 400, 700 nanometers, wavelength. Okay. It turns out some people can be colorblind. Maybe they have a deficient cone. The common one is red-green color blindness. It's an X-linked condition where you're missing or you have deficient red or green cone. And the genetics of it are easy to understand if you understand that if you're a male, your genotype for the sex chromosomes is XY. And if you're a female, your genotype just for the chromosomes is XX. We think of chromosomes in the human genome as pairs because we reproduce sexually. Okay, there's two sexes that come together that contribute the DNA. And there's 23 pairs of chromosomes in the human genome. However, the sex chromosomes, your sex is determined by one set of chromosomes, the sex chromosomes. The other 22 pairs are autosomes. And the reason why you have a pair is because you inherited half your set from mom, and the other set from dad. So, if you're a male, you inherited the Y chromosome from your dad. You have no choice but to inherit the X chromosome from your mom, although the two X's are different. if you're a female, you inherited the X chromosome from the sperm. Okay, so the sperm is either going to have the X or the Y when it fertilizes the egg, and so that's your genotype there. However, if you're a male and you have a deficient cone, say right there, colorblind man.
Does that mean they can't see any color at all, or is it some colors they can see when they're colorblind?
There's a test, and it's on my next slide. I'll show you. Okay. Yeah, but you're basically right. All right, so let's look at this cross right here. Colorblind man, normal, female, mom and dad, and they have 3 kids. Their daughter, when they kind of draw it half blue or half green, half red, what do they mean? Carrier. Are they colorblind? Well, what do they inherit? What would be the genotype? I used if they're a carrier, that means daughter must have received colorblind X chromosome from dad and X from mom. But because they have a normal X chromosome from mom, they're just a carrier. They're not colorblind. They're a carrier. Okay. How come the son's not colorblind? Both sons aren't colorblind. What's the reason based on genetics? What did they get from dad? If you're a son of a man who's colorblind, what did they get? I'm pointing to it. They got Y. Y is normal. It's X linked. Okay? That's why daughters, carriers, males, normal color. But now look at this cross. The carrier, normal male, and that cross basically is, let's see here. Okay, so they have daughter, also a carrier. That daughter is normal. They're trying to illustrate to you what are the odds the daughter will be a carrier. 50-50. Why is it 50-50? If you're a carrier, you have this genotype. You have kids. If it's a daughter, they'll inherit the X chromosome to you, but which one? It's 50-50. Okay, now look at the male. Colorblind son, not colorblind son. 50-50. Why is that? Those sons are either going to be XY or XY. If you get the X from mom, you're colorblind. The Y chromosome isn't going to protect you there. So, you know, that's that figure, study it. Okay, color blindness, inheritance. Many of us don't teach the genetics chapter. Well, I guess I'm just talking about myself. I don't teach it. But have some idea of genetics. The question was asked, what can you see or not see? Well, here's how you can tell. On this figure here, a person with normal color vision sees the number 7. Those who are colorblind usually can't see it. So this kind of answers your question. Can you see the 7? Those of you who can. What color is the 7? Green. Okay. Red-green, colorblind. People with red-green color blindness see either a 3 or nothing. Those with normal vision can see an 8. Okay, I do see an 8. But if you're colorblind, I guess these colors are a little off. You'd see a 3 because of the redness, red-green spectrum there. They'd be masked by all the green that's around it. It all look the same to you. Color blindness, it's pretty rare. But what I want to really focus on now is the the cell physiology of the photoreceptors, rods or cones. They happen to use a rod here. And there's two situations you should study, because you can have the lights on or lights off in a room. It can be daytime or nighttime. So we need to study both conditions and how the cell responds. I actually had a colorblind student, and I was using red tape and green tape on the lab practical, and I learned really quickly. Well, if I have a colorblind student, I can't use those colors. Okay, dark current, you're in the dark. The room is dark, absence of light, basically. Maybe there's a little light coming in. Dark current, what happens is, they call it dark current when you're in the dark. Basically, speak simply, can't see in the dark. Okay, you understand that? Okay, what's happening to the cell? In the dark, there's dark current. There's cyclic GMP gated channels that open. They allow cation influx. That's what's being shown there. So the thing to note, cyclic GMP gated channels open. So cations go in, like sodium and calcium. So I'll draw a little channel there. Sodium, calcium, influx. Okay, the cations go in. That's the dark current. Voltage gated channels open in synaptic terminal. Oh, well, I skipped a step here. That depolarizes the cell, that influx. You go from -70 to -40 right there. So try -40. Depolarizes the cell. -40. Okay, step 2. Neurotransmitter, number 2, voltage gated calcium channels open in synaptic terminals. When you open calcium channels in the synaptic terminal, that's a trigger for the neurotransmitter to be continuously released in the synaptic terminal. So that's 2 and 3. Number 2, V-gated calcium channels open in synaptic terminal. Because of that dark current. So think about that. You get an influx of calcium. Think of calcium as a second messenger, as a trigger to release neurotransmitter, step 3. I'll just abbreviate that NT, neurotransmitter. It's released onto the next cell, the bipolar cell. I'll draw little red balls to illustrate neurotransmitters right here. The bipolar cell is affected those neurotransmitters happen to be inhibitory on the bipolar cell. They create, remember when we learned about IPSP in lecture exam 4, they're inhibitory, okay? That stands for inhibitory postsynaptic potential on the next cell, the bipolar cell. That's what it says in the next step. Neurotransmitter causes IPSPs in bipolar cell, that hyperpolarizes it, basically turns it off. IPSPs in bipolar cell. Basically, it's turned off. Well, if that cell is turned off, there's no EPSPs in the ganglion cell, no action potentials, there's no signal from this cell. So therefore, you're not seeing whatever's in the room because it's dark. That cell's not on. So I don't want to keep consistent there. For step 5, basically they're just saying the bipolar cell is off. For step 6, they're saying there's no EPSPs in the ganglion cell. No EPSPs in ganglion cell. So I'm going to draw a ganglion cell. So there's no action potentials in the ganglion cell. There's the bipolar cell is off, so it's off. Ganglion cell off. No APs. Just right in here. I think that's what the last step says. There's no APs in the ganglion cell. So if the ganglion cell doesn't send signals to cranial nerve 2, you don't see it. It's dark. Okay. So let's turn on the lights. All right, step 1, in the light, now you can see things. Now photons of light are available. You close the channels. They're cyclic GMP gated channels. They were open in the dark current, now they close. Okay. So the cation influx stops. The photoreceptor hyperpolarizes. So what I'm writing is cyclic GMP gated channels close. We had that influx of sodium, calcium before. But with the channels closed, I'll put a big X through it. Without that dark current, there's no dark current. Without it, cell hyperpolarizes to -70. It was -40 in the dark current. But as it hyperpolarizes to -70 millivolts, that's basically what it's saying there. So now the cell, in the light, I'll change the 40 to a 70. In my picture. Step 2, the voltage gated channels in the synaptic terminal, they close. They close in synaptic terminal. With that, basically, you're going to stop releasing the neurotransmitters. Step 3, no NTs. So for my picture, I'm just going to erase the little red balls. There's no signal now, okay? So how does that affect the bipolar cell? Before, you were turning it off. Now, there's no IPSPs in the bipolar cell. The result is the bipolar cell is depolarized, it turns on. It's like a physiologist would say, you inhibit the inhibitory. Before, the photoreceptor was inhibiting the bipolar cell. But now with lights on, you inhibit the inhibitory. You've hyperpolarized it. No NTs. This bipolar cell is no longer off. Step 4 says lack of IPSPs on bipolar cell. I'll just say turn it on. They say depolarize, which is turning it on. I'll change off to on. Okay, now it's on, and what does it do to the next cell? Step 5, depolarization. Basically, it releases EPSPs on the ganglion cell. Okay. EPSPs on ganglion cell. That's what I'll say. So I'll draw neurotransmitters in the synapse there, EPSPs. So when you do that, the ganglion cell is now on, and you got APs, action potentials. Ganglion cell on, get action potentials. And on the figure, they illustrate the action potentials as individual red lines traveling down the axon. That is basically the visual signal sensory for cranial nerve 2. That's the cell physiology there. So those ganglion cells, I included this picture to show students that this axon, the fibers of the ganglion cell, they actually form cranial nerve 2. I tried to draw it before. On this picture here. Rods and cones, bipolar, ganglion. Those fibers form cranial nerve 2. They go right out the back of the eyeball. All the way to optic chiasm, optic tract, all the way to a nucleus and the thalamus, all the way back to the occipital lobe on the back of your brain. Well, anyways, I wanted to show you a detailed picture that's in the book that shows what's happening in terms of the photons of light activating the visual pigment shown on the left. So, I'm writing photons of light bind visual pigment. Yeah, that's what's shown right there, basically. And, what that does is it activates a transducin. Notice what happens to the skeleton key. Of the visual pigment called retinol. In the dark, the skeleton key would be bent. They call that the cis form, cis trans in the chemistry term. But when you bind the photons of light, notice it straightens out. And it's going to separate from the opsin. They call that bleaching. Well, anyways, it is shown here. Skeleton key straightens out. That activates the transducin, which is a G protein. Transducin will activate a phosphodiesterase PDE. So the next step, boom, you activate the visual pigment, you get transducin. That moves, G protein. It moves to activate PDE, a phosphodiesterase. And look what that does when it's active. It's going to take your cyclic GMP and convert it to GMP, getting rid of the thing that keeps the gate open on the dark current. So PDE is a molecule. That converts cyclic GMP to GMP. So when you get rid of the cyclic GMP, what it's showing you is that cation channel, it closes. So your cations, sodium and calcium, can't get in. The photoreceptor hyperpolarizes to -70. That was shown on the previous steps there. Okay, so that's basically that. So the key things are the transducin. After you kind of straighten out the skeleton key there, you activate that phosphodiesterase to eventually get rid of the dark current. No dark current. Okay, I'm going to move on from this slide. Another topic is light adaptation. Because you go from light to dark and dark to light, and there are adjustments that occur. And we've all experienced this, so it's easy to talk about. You can all relate to it. One thing to note is that cone cells are less sensitive. Rod cells are more sensitive to photons of light. Okay, if you're less sensitive, what that means is it takes more light to make you work. Okay? Rods are more sensitive. It's better for night vision. Because it doesn't take much light to make you work. It takes less light, you can still work. Requires less light to work. Basically, that's what it is. So, and when we, light adaptation is like, for example, the book that gave the example of you see a matinee. Okay, you go see the 11 o'clock showing of Endgame. I haven't seen it yet, so don't tell me about it. Has anyone seen it? You guys are studying too hard. You guys are good. Is it good?
Oh yeah, very good.
Okay, good. Can't wait. Anyways, so you see a matinee, it's dark, but then you walk outside, you know how bright it is? It's kind of that experience. So basically, what is happening there when you see that glare? That sudden rush of light, when you move from dark to light, you were set up for dim light in the movie theater. But when you walk out into the bright daylight, you overstimulate the photoreceptors. Bleach out is a separation of retinal from the opsin. When they say bleaching, I'm not sure why they use that term. Separation of retinal from opsin. Let's remember that opsin was the globular structure I drew. More or less like that. And then retinal was the skeleton key. Straightened out. Okay, and when it straightens out, it separates. That's called bleaching. Eventually, they'll get back together when you go back from trans to cis. So, when you see that white glare, there's compensations. Basically, you want to turn off rods when there's plenty of light. Okay? You see the white glare because everything's overstimulated. So I like what your book says. You go from dark to light. You see that glare. The adjustment is, they say for rods, turn them off. Transducin packs up in leaves. Okay, it goes to the inner segment. Transducin leaves. So I don't know if you remember the cell physiology we just looked at. Let me remind myself actually. Okay, when transducin is there, you don't got that dark current. But you know, if transducin leaves, this physiology can't happen. So you continue to have the dark current. That's basically like saying you turn the rods off. Okay, so that's this point here. The rods are off, transducin packs up and leaves. The less sensitive cones will take over. They're initially oversensitized, but will become desensitized as bleaching comes to an end. So the adaptation is transducin leaves. In other words, turn off rods, don't use them, there's plenty of light in the daylight. Less sensitive cones will operate. That glare doesn't last too long. You adjust after about a minute. The reverse situation is when you go from light to dark. Okay? You know, my dad's house is really dark. There's just no light. The house was built in the 70s. It's behind a brick wall. There's no sunroof or anything. So when on a bright day, when I go in there, it's like, I can't see. It's like a velvety blackness. I think that's a good description. When you go from light to dark, it's the reverse. See velvety blackness. You can see, but not well. Everything's just kind of dark when you go straight from a super bright room to a very dark environment. And so, in the dark, you turn cones off, there's not enough light. So, in the dark, the cones won't operate because there's not enough light. They're less sensitive. So you turn rods on. Basically, transducin returns. That takes a while, right? A few minutes. Transducin returns. Well, returns from what? It was in the inner segment. It goes back to the outer segment. Returns to the outer segment. Turn rods on. So even in the dark, when you've adapted, you still don't see well. You don't see sharp colors in the dark. You can make out edges, right? Because rods are not for color vision. The cones are. If you want to see the color of something and you can't see it in the dark, you have to turn the lights on. But you already knew that. Now you know why. You're using your rods or your cones. One of the last things I want to teach you is optic nerve, cranial nerve 2, the visual pathway. I kind of alluded to it when I taught split brain, case study where you have optic nerve receives information from both visual fields, right and left, illustrated blue, yellow respectively. The signals cross the chiasm, so that when you go to the lateral geniculate nucleus of the thalamus, which routes it to the posterior aspect or the occipital lobe, that's what you see. The left brain will see the right visual field and vice versa. Okay, now we've already established that. So what happens is each eyeball sees both visual fields. However, there's an injury to, well, you have a blind eye, say the optic nerve. There's a lesion or some trauma that's cut off. You lose both visual fields of that eye. However, you retain both visual fields in the other eye. You want to know what that's like, just walk around with the eye patch all day, okay? You still have information from both visual fields from both eyes. However, because the optic tract contains only information from the right visual field, this is what you would lose. Right visual field in both eyes. However, the left visual field is retained, seen by the right brain. Okay, so that's as basic as I make it. You can kind of Google this, it gets a lot more complicated. But it's the difference between if the optic nerve is damaged versus optic tract. This one, I don't know what it's like. I've Googled it, I've tried to search it, I've tried to look in the journals. What is this like? I have no idea. It's like if you look right down the room and this half is black. Okay, so I don't know what that's like. But it's very strange based on the wiring there, that's the case. Okay, well anyways, lesion in one place or the other, optic nerve, optic tract. When you come back from break, this is a picture of a student. Okay, I'm going to turn off the lights so you can see a little better. Oops. Okay. This is what your eyeball should look like when you're done, and I gotta check you off. Basically, the lab is simple, like the brain lab. I have a little half sheet here, you need one per group. List of 12 things to identify. This is not amenable to sticking with pins, so I just gotta go to you when you're done, you just show me the 12 things. Just go down the list, I check you off, and you're done. Big jelly blob, vitreous body or humor. Usually the jelly blob disintegrates and you just show me a puddle. Oh yeah, that was our vitreous, it came out. So dump it somewhere in a nice corner and keep it there and do not disturb it. See all the stuff they had to remove? Eyelids, extraocular muscles to get a nice round structure. Okay, remember, when you cut, use something sharp so it doesn't squirt when you puncture it. Okay. Anyways, to show me the layers, the outermost part, it's going to be what? The white. What are you going to call it? Sclera. Well, let me just go in order. Cornea, that thing. Pupil, that hole. Okay. Iris is the black around it. The ciliary body is also external to that. Okay, we call it the oral serrata. It kind of looks like that. What do you think that is? That's the lens. Okay, it's a pretty hard nodule once you get it from the cow eyeball post-mortem. Okay. Well, anyways, I call that vitreous body. Pay attention. The retina is very difficult for students. See that opaque, kind of beige, wispy layer that's over the black? That's it. Look for it. A lot of times, students get rid of it. It's like, where's your retina? And you forgot I said this, and I have to look around your tray. Oh, see that there that we're going to discard with all this junk? That's your retina. So be careful. It's difficult to spot if you don't know what to look for. But do you see how there's that blackish and iridescent layer? That's colon. Okay. Sclera, I pointed that out, the outer white part. The optic nerve. Do you see how the retina kind of gathers to a point right there? Basically, you poke a pin through that, on the other side you should see optic nerve sticking out. Maybe they gave you a centimeter or two, I don't know. Even if they gave you nothing, you should be able to see like a little dot. Oh yeah, that's right there. And optic nerve is a pretty easy one. So right around here, indicate, Dr. Wong, that's the optic disc where there are no photoreceptors. This one has a fovea. It's hard to see. It's a single black dot surrounded by yellow. Do you see that? That's hard to find. If you don't see it on yours, I'm going to give you a pass. Okay? But if you find it, I'll be impressed. It's hard to see. Okay? When we come back from break, we'll do that. Come back at about 9:20.